All right, so we're going to talk about the macromolecules carbohydrates. Okay, we just discussed the different types of reactions that can occur with macromolecules, such as dehydration and condensation and hydrolysis. And that's how we create larger polymers, like we mentioned in the last video, of these macromolecules, such as carbohydrates. Um, so carbohydrates are broken up into three categories. Okay, so we have monosaccharides. So all carbohydrates contain elements of carbon, hydrogen, and oxygen in a ratio of a 1:1. There are three groups of carbohydrates: monosaccharides, disaccharides, and polysaccharides. The equation will look something like this: CH2O, okay, and then you're going to multiply this whole equation by whatever the number might be, okay? And I'll show you an example of that. So for monosaccharides, this is our energy source. Some examples would be glucose, ribose, fructose, galactose, and we'll go through some of those later on. Disaccharides, also an energy source: maltose, sucrose, which is table sugar, and lactose. Each one of these disaccharides is created via a dehydration synthesis reaction of two monosaccharides. So di meaning two, mono meaning one, okay? And then we also have polysaccharides. Okay, so polysaccharides are multiple monosaccharides linked together. So this would be your polymer of carbohydrates. So we have monosaccharides, which are monomers, and polysaccharides, which are polymers. These are energy storage. Okay, so they're not really necessarily used for energy, as you have to break apart the monomers in order to use those molecules, or such as glucose, for energy. So these are usually meant for energy storage. So our bodies kind of take in the glucose from our blood and they store it in a polymer chain called a polysaccharide. So their energy storage, structural support, and cell-to-cell communication. Glycogen, starch, cellulose would be examples of polysaccharides. Okay, so again, the monomer is the actual energy source, and we store it in our bodies, or like say our muscles, as polysaccharides such as glycogen. Okay, and they act as a storage molecule. So there are a number of different ways we can look at monosaccharides. Monosaccharides can have carbon chains of three, and we call those trioses. We have carbon chains of five. Sorry, not carbon chains. These are n equals three. So remember, in the equation here, we had our n right here. So this would be n trioses. So carbon chain three it could be. But our n equals three. So what I would just simply multiply this equation by n, which is three. We can have pentoses, which is five, such as ribose or deoxyribose. These are used for our RNA and DNA. And then we also have hexoses, which would be six. Okay, so again, for trioses, we multiply the whole equation by three, giving us C3H6O3. Pentose would be C5H10O5. Hexose, C6H12O6. And all I'm doing is simply multiplying this equation by those specific numbers, 3, 5, and 6, giving us the different structures of monosaccharides. So the first monosaccharide that we're going to talk about, and we're going to probably talk about this one quite substantially over the next semester. This is the glucose molecule. Glucose is the most important carbohydrate fuel in human cells. We use it through cellular respiration, where we combine glucose with oxygen to create ATP, or usable energy. There's a number of different reactions that we're going to learn that helps turn this molecule into usable energy for our cells. We store this in our muscles, and it's circulating in our blood. And when we look at our blood sugar level, so when diabetics are looking at blood sugar level, they're looking at the glucose level within their blood. Okay, this is also a hexose, and it's found in cells as energy. Okay, so we have a linear structure here, and if we count from carbon 1, 2, 3, 4, 5, 6, so we can see it's a hexose. There's a number of different functional groups on this monosaccharide glucose. So we have a carbonyl or carbonyl group here, okay, on carbon 1, and we have a number of different hydroxides, with carbon 3 having a hydroxide on the opposite side. So what happens is this is when glucose is in its dry form, okay, so it hasn't been exposed to any water yet, okay, but in our cells, obviously glucose goes, interacts with water. So what happens is you get a bending of the glucose chain. So what happens is this carbon 1 starts to progress down here this way, and this carbon 6 starts to move up this way. So 1 moves down here, 6 moves up here, and you get a structure that looks like this. Okay, 6 is all the way at the very top, 5. So what happens is the OH on 5 actually, what happens is the oxygen, because this carbon becomes has a partial positive charge because of the double bond to the oxygen. Oxygen is hogging all the electrons for this bond. And what happens is this oxygen actually starts to become attracted, and it's called a neutrophilic attack. And that's something you learn later on in university. Maybe you learned it in grade 12, but it's a neutrophilic attack on this carbon. So this oxygen is attracted again to this specific carbon, causing this bond to occur right here. Now, this is the end product, the cyclical structure of glucose. And this is the structure that we're going to be experiencing or looking at and a lot more throughout this course. Yes, you can look at it this way. However, this is what you're going to see for the most part. Okay, this is something that you're going to need to learn how to draw and know how to draw, and I'll walk you through a couple of steps in order to teach you how to draw that in a second. Okay, so again, this oxygen is attacking this double bonded carbon due to the fact that, again, this carbon is slightly charged, has a slight positive charge because the oxygen here is taking the electrons or hogging the electrons in this bond. Okay, so it's attacking this carbonyl group, this carbon with the carbonyl group. We also have a monosaccharide called fructose. This is also a hexose, if you can count 1, 2, 3, 4, 5, 6. It's found in fruits, and it actually, instead of having the carbonyl aldehyde group, it actually has, the carbonyl aldehyde group like the glucose. It actually has a ketone group right here with a number of hydroxide. Okay, and it also too, it is linear when it's dry. However, when it's in our cells, what happens is that it interacts with water. And again, the hydroxide group on 5, so it's the same type of rotation that happens here. 1 is going to come all the way down this way, so it's going to draw, you can see here, draw 90 degrees. This one is going to move up to here. Okay, so 6 is going to come up, 1 is going to come down cyclically. Okay, and what's going to happen is that the OH on 5 is actually going to, again, have a neutrophilic attack on this carbonyl group right here. Okay, again, because this carbonyl group, that carbon is now partially charged positive. Okay, and this oxygen is, because it's electric negative, it's going to create that neutrophilic attack on this particular carbon. Okay, and once that particular carbon bonds with that oxygen, that carbon now can let go of one of the bonds to the oxygen, to the double bonded oxygen here, can let go of one of the bonds of the oxygen, ultimately gaining a hydrogen proton from the water, usually a hydronium atom, bumps into it, takes that hydrogen, and now you're left with a fructose molecule like this. Okay, again, fructose is found in sugar, and it's another important monosaccharide, but again, glucose is the most important polysaccharide. Okay, so just so you know that it does, when it's dry, there hasn't been exposed to water, it looks something like this. However, once in our cells, this is what it kind of looks like, is this cyclical structure here. What we notice about the difference between fructose and glucose is that the cyclical structure of fructose is actually made up of 1, 2, 3, 4 carbons and an oxygen, okay, where glucose is made up of 1, 2, 3, 4, 5 carbons and 1 oxygen. The sixth carbon being up here for glucose, and the 1 carbon being down here for fructose, and the 6 carbon being up here for fructose as well. So we also have the monosaccharide ribose, and ribose is a pentose, so it's only a 5 carbon chain. So we have 1, 2, 3, 4, 5. Okay, and we see it has an aldose group up here, so it has a carbonyl aldehyde group up here with a number of different OHs. And the cyclical structure of this is exactly this right here: 1, 2, 3, 4, 5 carbon ring here. This particular monosaccharide actually forms the backbone of all of our DNA and RNA, and we'll learn about that when we talk about nucleotides in the next few lessons. Okay, so monosaccharides. So again, the main one that we're going to be talking about is glucose. Glucose has actually 2 forms. It has a alpha form, so alpha glucose, and a beta form called beta glucose. And realistically, what you notice here is that there's a difference in the two. And the only structural difference is the OH, the location of this hydroxyl group here. Okay, the hydroxyl group on carbon 1 is pointing down in alpha glucose, and the hydroxyl group on carbon 1 in beta glucose is actually pointing up. Okay, although this may seem very minuscule to you, it actually have completely different functions because of those differences. They do possess the exact same chemical formula. So if we were to count up all the carbons, all the hydrogens, and all the oxygens, they would have the exact same chemical formula. However, their functions are going to be a little different, or a lot different, depending on depending on the orientation of this hydroxyl group. Okay, so I'm going to show you how to draw the alpha glucose molecule right now. Okay, so in order to draw the alpha glucose molecule, I am going to first start off with my oxygen. So step 1 is to start off with my oxygen. So I'm going to draw my oxygen. Sorry, my pen, this is not a very, it's not going to be a very pretty drawing, but I will do my best. Okay, so I'm going to start off with the oxygen. I am then going to draw a hexagon. 1, with my oxygen there. Each vertice of the hexagon represents a carbon. Okay, now I'm going to draw carbon off of carbon 5. So I'm going to draw carbon off of carbon 5. Okay, I'm going to add my 2 hydrogens. So I got my carbon here. I'm going to add my 2 hydrogens in my OH group off of this carbon. Okay. And now I can start going into my hydroxide group. Again, this is alpha glucose. Okay. So now I'm going to start going into drawing my hydroxides. Okay. The hydroxides in alpha glucose actually follow a down, down, up, down rule. Okay. So my carbon 1 is going to have a down hydroxide. Okay, again, this is alpha glucose. My carbon 2 is also going to have a down hydroxide. My carbon 3 is going to have an up hydroxide. So I'm going to draw up here. My carbon 4 is going to also have a down hydroxide. Okay, now I can just simply fill in my hydrogens. So each carbon is going to have a single hydrogen, 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 and hydrogen. Okay, you do need to know how to draw this. Although that's not a very pretty picture of the glucose molecule, it is the alpha glucose molecule. This is just the alpha glucose. You're going to also need to know how to draw the beta glucose molecule, and you can follow the same steps. Okay, however, with the beta, give me a second. With the beta, okay, again, instead of having that down hydroxide like we just showed, you're going to have an up hydroxide on carbon 1. Okay, So you can write this down. Okay, for alpha glucose, you're going to follow a down, down, up, down principle for your hydroxide. So down, down, up, down. For beta glucose, it's going to be up, down, for beta glucose. You will need to be able to draw both of these. So make sure that you know down, down, alpha glucose, up, down, beta glucose. Okay, that, like I mentioned before, they do have the exact same chemical structure. However, their function is going to be a lot different. And that makes them isomers of themselves. Okay, so because they're isomers, they do have the same formula, like I mentioned, but again, they do have a different structure. This changes the function of the molecule. So just because that OH, that hydroxyl group is facing up on the beta, it actually changes the function of that particular glucose. It may not seem like a lot, but it does. And a prime example of that is back in the 50s, there was a drug, phthalimide, that was an anti-nausea drug for pregnant women. And what happened is this was the chemical structure of phthalimide right here. And however, when women would actually take the phthalimide drug, what would happen is that it would interact with something in their system, causing it to kind of mirror itself. Okay, so this amine group became different, causing the shape and the actual form of that drug to be different. Okay, and what would happen is that these pregnant women that would take this for anti-nausea would actually, they would actually give birth to babies that had no limbs. Okay, and this is an example of some of the limbs that they had made up in the 50s, 40s, and 50s for these babies. These babies were born with no legs, no feet, really, really short arms, no arms, no hands. It was pretty sad, and that's because it formed that specific isomer when interacting with something in the woman's body. So again, we have those monomers. Those monosaccharides, and now we go to the disaccharides. And disaccharides are composed of 2 or 3 simple sugars. So 2 or 3 monosaccharides attached by covalent bonds. And these covalent bonds are called glycosidic linkages, which are formed by a condensation or dehydration reaction. So right here we have 2 glucose molecules, and you know that the OH is down. So these are alpha glucose molecules. Okay, so what's going to happen here is the hydroxide group of 1, the hydroxide group of 1, it doesn't really matter which one of the hydroxide group, it changes, I guess, but the hydroxyl group of 1 is going to interact with the hydrogen of another, okay? So glucose, 1, 2, 3, 4, carbon 4 of one atom, of one molecule, is going to interact with carbon 1 of another, okay? And you're going to get the formation of water. Water is going to come out, so it's going to be condensation, and you're going to get this glycosidic bond here, okay? Glycosidic linkage, where they both share that electron. Carbon 1 on one molecule and carbon 4 on another molecule share this oxygen, okay, forming a glycosidic linkage. Alpha glucose to alpha glucose forms a alpha glycosidic linkage. Okay, so disaccharides contain the same functional groups as the monosaccharides. Therefore, they are hydrophilic. Okay, and you can still see here, right? You still have all these hydroxyl groups within each one of the monosaccharides. Okay, so again, the hydrogen interacts with the hydroxyl of 1, and you get the formation of water and a glycosidic linkage. Alpha glucose with alpha glucose forms a maltose, and when the bond is coming down, what happens is you have that alpha. When the bonds to the oxygen are coming down from carbon 1 and carbon 4, like such, you're going to have an alpha glycosidic linkage. Okay, you should know how to draw this whole thing right here. Okay, again, this is alpha glucose. We already talked about how to draw that alpha glucose, and you just need to get rid of H2O, so 1 hydrogen, 1 hydroxyl, and you get this alpha glycosidic linkage. So we also have glucose and fructose, which form a disaccharide sucrose. This is the only reason I have this here is because this is our table sugar that we use. If we look at again the chemical linear structure of glucose and fructose, glucose right here we have an aldose group here, and right here we have a ketose group here, or a ketone and aldehyde carbonyl. They're both 6-carbon ring hexose. They One has aldehyde, one has ketone. Fructose is found in fruits, lower glycemic response. So what happens is fructose actually goes into the liver and gets converted to glucose. It takes a long time for the fructose to transfer into glucose, and our liver has enzymes and pathways that we'll talk about that turn fructose into glucose. Fructose is unusable to us as energy. Glucose is the usable energy. Okay, so we don't want fructose circulate. Well, we don't need fructose circulating in our blood. So what happens is the fructose goes to our liver. Our liver goes through a process of gluconeogenesis, which converts the fructose into glucose. And then it's released back into our blood and taken up by our cells and stored by our cells as glycogen. Okay, sucrose is formed also by honeybees and fructose, not sucrose. This should say fructose. Honeybees and other fruits as well would have fructose. But again, sucrose is a disaccharide. So we do have a number of different types of bonds for disaccharides. We have two types of bonds for disaccharides. Remember how I said this was an alpha glucose? Alpha glucose goes to a maltose, and that creates a alpha glucose alpha 1 to 4 linkage. Okay, so carbon 1 of one to carbon 4 of the other monosaccharide forms a 1,4 alpha linkage, okay, because this is two alpha glucose molecules. But what happens is if we have a galactose, let's just say, a galactose and a alpha glucose, what happens is the hydroxides, the hydroxyl groups on either or on opposite ends, and you get a beta glycosidic linkage. And this beta glycosidic linkage, this is lactose. Okay, this beta glycosidic linkage is a lot different than this alpha glycosidic linkage. You need to know that you don't need to be able to draw the beta glycosidic linkage. However, just know the difference, that there is a difference because of that. So this will come up and the bond will be a lot different in form. Lactose is actually a sugar or a disaccharide that our bodies sometimes can't break down. Okay, so we need to break out down. When we take in milk, milk has lactose in it, which is a disaccharide. Lactose is broken down by an enzyme in our bodies called lactase. And for those of you that are lactose intolerant, what happens is people that are lactose intolerant are missing that one specific enzyme, lactase, and they cannot break down the lactose molecule. So it causes a lot of stomach issues. Okay, but this is known as a beta glycosidic. That's when you have an up hydroxyl group and a down hydroxyl group interacting with each other to form this specific beta glycosidic linkage, such as lactose. Where alpha, when you have two alpha glucose molecules, you get an alpha linkage, 1,4 alpha linkage. This would be a 1,4 beta linkage, glycosidic linkage. Okay, so now we have our polysaccharides. Again, our polysaccharides are made up of a number of different monomer subunits. Okay, so they could be a number of different glucose molecules, a number of different, glucose, beta glucose. It could be a number of different fructose and glucose put together. It could be, something completely different. And that's our polysaccharides. And we'll talk about the one that's going to be different as it has a bunch of amine groups in a second. But anyways, our first one is amylose. Okay, this is used as storage as plants. This is used as starch storage in plants. Sorry, okay, and it does alpha glucose. Okay, a bunch of alpha glucoses. Okay, monomers that make up these alpha 1 to 4 glycosidic bonds. Okay, again, the carbon 1 on one molecule to the carbon 4 on another molecule forms this alpha 1,4 glycosidic bond. Okay, and we know that because they're both pointing down here. We also have amylopectin. Amylopectin is also made up of alpha glucose molecules. Okay, it has an alpha 1,4 linkage in a chain. So similar to an amylose, it has the 1 to 4 linkage and it forms a chain. However, amylopectin can actually form a 1,6 branch as well. Okay, and you get a 1,6 glycosidic bond here, where carbon 1 actually interacts with carbon 6 on the, carbon 1, the oxygen on carbon 1 actually interacts with carbon 1 interacts with the oxygen on carbon 6 in this particular polysaccharide. Okay, so you have an alpha 1,4 linkage and an alpha 1,6 branch. Okay, and as you can imagine, the 1,4 is linear, and then the branch comes up here. And you can also have, and then it branches up here, and then maybe one goes up here and another one. So amylopectin can be not only linear chain, but it can also be branched because of that 1,6 branch. We have glycogen. Glycogen is made up of alpha glucose. Okay, it is an alpha 1,4 linkage in chain as well as an alpha 1,6 in branches, similar to the amylopectin. And this allows just for more storage of glucose molecules. This is for energy storage in humans or in animals. So in our bodies, in our muscles, let's just say, our muscles have a lot of glycogen readily available to be used, to be broken apart, so that they can be used in, say, when you do an exercise, when you go for a run or whatever, for whatever reason. Okay, so this glycogen right here is made up of alpha 1,4 and alpha 1,6 branches. We also have cellulose. So cellulose is made up of beta glucose and alpha glucose combined together. It is a beta 1,4 linkage because, again, you have that 1,4 going up, but you also have some of those alpha branches going down as well, depending on whether it's alpha beta, or beta beta glucose. Okay, so this is for cell walls. It's the formation of cell walls, and it is insoluble to digest. So that's what cellulose, we can't digest cellulose in humans. So that's the roughage that people say you need to eat. Then we have that one that I was talking about with the amino groups. Okay, we have chitin, and this is made up of N-acetylglucosamine, which are these molecules right here. So it's a glucosamine molecule attached to an amine group, okay, with a carbonyl or a ketone here. This is made for exoskeletons of, like, say, ants or other insects. So this is what that is made up of. So cellulose is very, very strong. This is due to the formation of hydrogen bonds resulting in tight bundles called microfibrils. These intertwine to form material that is tough and insoluble. Okay, so humans are unable to digest cellulose. They lack the enzymes to break the beta 1,4 linkages. So that's the reason why we can't digest cellulose. Okay, not only that, but you can see the hydrogen bonds forming in between each chain. Okay, and it kind of intertwine as they go into a single, into a plant cell. Okay, so despite the cellulose is an important, despite the fact that we can't, digest the beta 1,4 linkages, it is an important part of our diet because it gently scrapes the walls of our intestine. Remember how I said it acts as roughage, and it causes mucus secretion from our intestines, which lubricates our feces to aid in waste elimination. So it's important to eat that cellulose and eat those vegetables that have a high amount of cellulose. It also helps keep feces moist and binds water in the large intestine. So again, it brings that water back into the large intestine, so you're not, so your feces are nice and healthy. Is really what it does. So these hydrogen bonds again allow for cellulose to be extremely strong, but also our bodies cannot break down the beta 1,4 linkages. And that's it. Next lesson will be on lipids. Thanks.